Welcome to the second lecture of the Marine Community Ecology course, and today we're going to go through a brief overview and history of community ecology. We're going to start with just a couple of the drawings that I've asked you all to do. There were a whole bunch of kelp forest drawings. We had some deep sea vent communities, a zooplankton community, a marsh community, and a sort of sandy coastal channel community. And another drawing was this one. This one actually isn't by a student in the class. This is by a guy called Alexander, and the one I'm referring to is Alexander von Humboldt. Alexander von Humboldt was, for all intents and purposes, probably the first community ecologist we know. This is him on the left, and he has his statue in Central Park because Alexander von Humboldt actually spent most of the time, even though he was German by heritage, he spent most of his time in the Americas and ultimately became a citizen of Mexico. The drawing I showed was a drawing of the Chimborazo volcano in Ecuador. And what Humboldt did is he sort of hiked up the Chimborazo, which was quite the technical feat at the time because it's quite a high mountain. I think it's about 18,000 feet. On the way of his hike, while he was suffering from altitude sickness and everything, he sort of sketched out the different zones and different communities on the Chimborazo. This is an actual photo of the Chimborazo on the left, and some of these really cute sort of cushion plants and succulent communities that you get on this mountain. And if we zoom in on the drawing here, he sort of just used different genera of plants to sort of define some of the zones that he observed. And you can see that he sort of had some of these altitude measurements in here from 4,100 to 4,600 meters, and so on and so forth. Now, one of the reasons I quite like Alexander von Humboldt is because he was very much ahead of his time as a scientist. He was born into quite a lot of wealth in Germany, but as he was exploring the world in the Americas, he became a very staunch advocate against slavery, which was raging at the time, against colonialism, and he was very outspoken about it. He was almost certainly also gay, which of course was punishable at that day and age, and he had this really deep understanding that whatever we're observing in nature really is all connected, from sort of the physical sciences to geology, earth science, botany, geography, zoology, and so on and so forth. Everything sort of flows into each other to create ecological communities. And that's really the crux of it, because ecology is the science of everything. It derives from oikos and logos, which are Greek words for everything and science. So it is the science of everything. And that really sort of goes back to this central domain that we observe communities to be if we sort of scale life on our planet from the cells to the entire planet. Now, one thing to consider here is, especially with plants, obviously the plants at the very bottom of the Chimborazo would never really ecologically interact with the plants at the very top. And that really puts the issue of scale back center stage. And it helps us to think about this when we think about how we want to quantify ecological communities. We can do so at a lot of different scales. We can do so at local scales, at regional scales, or at global scales. The Chimborazo obviously would fall under the local scale, and even though the plants at the bottom would not interact with the plants at the top, they would still sort of qualify as a single community. We could also expand that to, for example, the entire Andes or the entirety of South America, and then of course we could do the same thing at the global scale. So let's talk a little bit about how we actually quantify ecological communities. The first thing we might do is we might look at these two photographs here of fishes on a coral reef, and the very first thing we could do is simply count the individuals. And counting the individuals gives us a metric of a single community that we call abundance. So that is simply the number of fish you see in these circles. Abundance is always composed of integer values, so they are whole numbers between 0 and positive infinity. There are no half fish, there are no negative fish. It's got to be 1, 2, 3, 4, 5, and so on and so forth. Now we can change a few things on this. The first thing we can change is we can populate this community with different species. So now on the left side you have a green fish, a white fish, and a blue fish, and on the right side you just have the blue fish, which means that our species richness, which is our estimate of how many species there are, has changed. On the left we have 3 species, so our species richness is 3, and on the right we have 1 species, which means our species richness is 1. Similar to abundance, species richness are also always integers. There are no half species, there are no negative species. It's got to be full numbers between 0 and positive infinity. Okay, so now I've changed the species richness in the second community as well. I now have 3 species in that community too. But as you can see, the distribution of species is different. So on the left side, we have fairly even spread between those 3 species. On the right side, we only have 1 of each of the 2 new species. And this gets to a metric that we actually call diversity. I will most likely use diversity in the context of species richness as well. But true diversity actually takes into account the abundances of different species as well. Now, diversity can be calculated using a variety of different indices. I've given an example here. This is the Shannon Diversity Index, which is usually denoted with the letter H. You don't have to really worry about the formula. There's code that can do this for you. And what you get are these values here, 1.0986 and 0.8675, which suggests that diversity is higher in the first community, right? Because we have a more even spread of the fish across these species. But the important thing to note is that in and of itself, the diversity index doesn't actually tell you all that much. It's mostly a relative indicator of the species richness and abundance distribution, or what we call evenness, across these 2 communities. So these can be don't have to be integer values. They are index values that are simply positive values, but can take any sort of decimal within those numbers. Okay, so now I might go ahead and take that white fish in the very bottom right here and change it to a different species. And now we have a community that has the same abundance, it has the same richness, it has the same diversity index, the Shannon, but they are very clearly different. And that gets very tricky to quantify. Because for all intents and purposes, they are the same. They just differ in what we call the composition. So community composition then refers to differences in species composition. And composition can also change based on the abundance. So these 2 communities have the same richness and they have the same abundance and they have the same species, but their composition differs in terms of the relative abundance of each species in the community. The major issue with comparing communities is that there is no quick and dirty way of doing it. There is no simple index that we can use. There is no simple calculation, no simple metric. Community composition, we can only compare across several communities. So community composition can't be calculated for a single community. And we usually have to use some slightly more complicated multivariate techniques to do this. Okay, since we're now comparing multiple communities, this is also a good time to introduce a couple more terms, a couple more metrics that we tend to use in community ecology. The first one's called alpha diversity. So this is the local scale diversity that you're observing. So if you're quantifying a single community, such as, a community of fish on a certain reef or a community of birds in a certain pond or whatever, that is alpha diversity. So that would be the number of species in that community. If you're doing that across several communities within a region, that is called gamma diversity. So the total number of species in that community would be the gamma diversity of that system. And then the one in between refers to species turnover. So this is called beta diversity. This is the turnover of species across these different communities. So in a sense, it's a metric of how different these communities are in terms of the species that exist in these communities. So again, you see that this goes towards the composition of those communities in terms of their species. All right, to make this a little less abstract, we can sort of conceptualize this in a table, perhaps, rather than just these circles with fish in it. So if you have 3 sites, site A, B, and C, with different communities, we usually sort of denote this with a table where the rows are the different sites, and then we have species as columns. So you can see if you're going to site A, you have the green species. Right here, I'm just doing presence absence. I'm not doing abundance yet. So the green species is present, so it gets a 1. The blue species is present, so it gets a 2, gets a 1. The orange species is present, so it gets a 1. And then the white species isn't present, so it gets a 0. Same for B, same for C. And then we can use abundances as well. So we can count out those species 1, 2, 3, 4, 5, 6, 7. So at site A, species 1 has 7 individuals, and so on and so forth. And these tables are quite useful. They're going to be the main sort of way in which you quantify these communities. And obviously, you can tell there's a lot of sites, there's a lot of species. That's a dimensionality problem that we'll get to later in this lecture. Okay, so there's a couple more things, couple more properties of communities that we can quantify that are a little more difficult or a little more complicated. We call these second order properties. One of them becomes quite obvious when we look at these 2 communities of reef fishes. So the one on the right has mostly 1 species. It's sort of big, it's silvery. And the community on the left has lots and lots of really small fish species. They're colorful. So very clearly they differ in that one community has tiny and colorful fish and the other community has large and sort of plain silvery fish. These are what we call traits. So each species has certain biological traits, and we can use these traits to basically biologically differentiate communities, not just based on their taxonomic identity, so not just calling them a certain species, but also trying to figure out, what is it that makes these communities different in terms of the biology of these species. And then the second thing that we often do, and that works with traits as well, is we can look at the environmental drivers of these community, and that's called species-environment relationships. So you're trying to understand the links between the environment, certain characteristic of a site, and the species that are present. Okay, this is just basically a little cheat sheet that is in the presentation that you can go back to at any point in time. So we have our first order variables, which are abundance, species richness, diversity or evenness, and species abundance distributions. We didn't really talk about those, won't worry too much about them. They're all quantifiable for a single community. So I can take a single community and I can get a value for these metrics. And these are what I and most people generally refer to as the community structure. So species richness and abundance, community structure. Community composition is usually denoted as compositional differences, so differences in the types of species that are around and they can only be compared if you have multiple communities. So it has to be compared across a set of different communities. It doesn't work for a single community. There's no single value of composition that you could derive for a single community. And then our second order variables are the trait, diversity and composition, and the species-environment relationships, which consider the biological traits of species and the characteristics of the environment. Okay, now armed with that knowledge, we can go ham on comparing as many ecological communities as possible. We just got to find the right values, the right tools to do so. And this is, the very sort of complicated rundown of all the different things you can do. Trust me, there's a lot of different ways to do comparisons in community ecology. The truth is, oftentimes they yield very similar results. So whether you do Pelu's evenness index or the Shannon-Wiener index or the Simpson's diversity index, oftentimes the results are probably going to be mostly comparable. But we'll see sometime later in this course that sometimes which multivariate technique you're choosing, for example, does matter quite a bit. All right, and that's sort of the basic, those are the basic tools you'll need to compare ecological communities. You can now take a couple of minutes to think about species richness in particular. So this is, again, the number of species in a community. And think about what influences species richness at a global scale. Where do you think, especially in the marine environment, do we have concentrations of species, and why do you think that is? What are the drivers or causes of species richness in the marine environment? And we'll start by talking about estuarine fish. So we're here at UTMSI. We have a big estuary, subtropical estuary, so this is very close to home. And we're going to look at the global distribution of species richness in estuarine fish communities. And this is a map from Vasconcelos et al. In 2015. The colors here are somewhat non-intuitive. Usually we use those heat map colors, green to red, to indicate actual values. Here they are just the different regions, and the number of species is denoted in the size of these circles. And what you can see is that up here, for example, in the UK, the circles are very small, which means that there are very few species, which makes sense because nobody wants to live in the UK because the food is terrible and they have warm beer and shitty weather. Whereas we see a concentration of really big circles over here, over here, over here, and over here. And when they looked at the drivers of species richness, they found that biogeographic region was really important. Sea surface temperature, the warmer it got, the more species. Estuarine area, so the larger the estuary, the more species. And connectivity was also really important. Connectivity means how connected a system is to other systems or the open ocean. So this just basically refers to how much interchange there is between that estuary and others. We can do the same thing by looking at coral reef fishes. So now we've moved on from the murky inshore waters to clear tropical waters with coral reef fishes. Obviously, coral reefs are somewhat restricted to the tropics, so we don't necessarily go beyond a certain latitude. But if we look at this particular map, we can see this time the red actually denotes hot colors. So these are hot spots, lots of species where we have the red colors, and much fewer species where we have the blue and green colors. And we can see that there's a concentration, massive concentration of species richness in this area that we call the Indo-Australian archipelago, or also the Coral Triangle. So this triangle right here has the highest species richness. And perhaps not surprisingly, they also looked at the relative influence of different variables on species richness, and they found that coral reef area, biogeographic realm, sea surface temperature, and connectivity were important drivers of coral reef fish species richness. Let's switch gears to a non-vertebrate. So let's look at cephalopods and their biodiversity on a global scale. This is a very similar map. Again, we have a heat map where the red denotes higher species richness, and this should look somewhat familiar. So again, we have the coral triangle and the Indo-Australian archipelago right here, and we can see that right here is sort of the very center of diversity with most cephalopod species right here. The one thing to highlight is that there seems to be a fairly warm color right here in the Mediterranean. And that actually ended up being sort of a phylogenetic quirk. So the authors identified phylogenetic history as an important driver of global diversity patterns because there were simply some lineages in the Mediterranean that seemed to have diversified more than one would have expected, which sort of created that little hotspot right there. And then, of course, region turned out to be an important factor again as well. We'll have one final example, and we're going to look at tunas and billfishes, so large oceanic predators, big blue fish that spend their entire lives in the oceanic environment. And this is what their map looks like. So again, red denotes higher species richness, blue denotes lower species richness. And what we can see is that the Indo-Australian archipelago right here, that is the center of diversity for other taxa, doesn't seem to be that hot for species richness. Instead, we get these bands right here, almost right here in the southern Pacific, in the northern, or the about 20 degrees latitude in the North Pacific, here in the Indian Ocean, up here, and off Australia. And what the authors found is that there's actually this sort of hump-shaped relationship between diversity and temperature, where temperature has a positive effect on diversity until a certain degree. Oops, excuse me. And then it decreases. Okay, so now let's look at all different taxa. This is a sort of holistic, comprehensive examination of all sorts of different marine taxa and their diversity globally. And you can see that coastal fishes, yeah, that pattern looks familiar. Non-oceanic sharks, yeah, pattern looks familiar. Non-squid cephalopods, we've already talked about some of those. So all the coastal taxa are on the left, and all the oceanic taxa are on the right. And we can see that there's quite a bit of difference in these maps in terms of what they look like, which makes sense because some of them live obviously in the open ocean, some of them live on the coast. But what's perhaps most important is that we see this clear increase with temperature for coastal diversity. And we see again this sort of asymptoting power function of diversity with temperature for the oceanic taxa. Now, the question is, why? Why is that? I'll give you a minute to think about it before I move on, or you can just pause this. So the answer is most likely related to the fact that in the open ocean, the tropics become a desert. So there aren't a lot of nutrients to go around, and solar irradiance is really, really strong, which means that at the equator and around the equator, there isn't a lot of food in oceanic environments. And on the flip side, there's a lot of costs to existing there because temperature is really high, so your metabolism is really high, which means that you're probably expending a lot of energy and you can't find a lot of food. Whereas in coastal systems where you have nutrient input from terrestrial matter, you get a lot of rain that washes down nutrients from islands and the continents. That nutrient limitation probably isn't as much of a problem. And that's probably why we see that difference in diversity patterns between the open ocean and the coasts. And then there's another quirk here that I want to alert you to, and that's the pinnipeds. So those are your seals and sea lions. And they show something very different from all the other taxa in this column right here. And the question is, why are pinnipeds most diverse in temperate and polar regions when all the other taxa, coastal taxa, don't seem to do that? So you can think about this, you can pause the presentation, think about it a little bit, but I'll reveal it right here. There was a great paper by Grady et al. In 2019 that was published that sort of made the link between physiology and this advantage of predatory marine animals that they may have. So the argument is essentially that mammals and birds that can regulate their body temperature are at an advantage over fishes that can't regulate their body temperature in really cold environments. And that's why they conjectured that that mammals and birds are so successful in polar environments, and that may drive some of these diversity patterns that we see in pinnipeds compared to everything else. Okay, so we've talked about some of the global patterns in diversity, but a lot of community ecology doesn't happen at the global scale, because guess what? Doing stuff globally is really, really difficult. It's really expensive. You need a lot of data from a lot of different places. Doesn't happen all that often. So most of the time we're really sort of looking at a single community like this coral reef right here, and I might look at these 4 different fish and be like, why are you here? What's the deal? Are you a community? What are you doing? And as most fields, community ecology has gone through a lot of different stages in terms of what people think about communities. And this is the ideal scientific journey. You start by knowing not a whole lot. Someone has a brilliant idea, someone has a better idea, and then someone says, well, they're all wrong, and I have a much better hypothesis, and so on and so forth, until eventually you sort of start to derive a general picture. And in community ecology, this, of course, started by simply observing patterns. And the first person to really do that, I've mentioned him before, was Alexander von Humboldt. He was, at least in my books, the first true community ecologist who thought about ecological communities in a fairly holistic way. Another scientist, early scientist who thought about communities a lot was a guy called Frederick Clement. He wrote some page turners like the Phytogeography of Nebraska, which I'm sure is riveting if you open it up. And he thought of ecological communities as these superorganisms. So a distinct community that almost functions like its own little organism. And we can conceptualize this perhaps a little more scientifically by thinking about species distribution. So he basically thought that, okay, we have a community here that aligns with all these species distributions. So this community here forms a superorganism, and the other one did as well. And we can kind of see that in nature sometimes. So for example, if you think about a mountain range, you can see, oh, there's this forest community right here that is very much connected. It's the same species that form this community throughout. There's obviously a lake community of phytoplankton, and so on and so forth. But a guy that sort of challenged this and thought very differently about community was Henry Gleason, who also had some very pragmatic work, such as Species in Area. Wonder what that is about. But he thought of ecological communities more as a conglomerate of species with individual tolerances. So very contrasting to Clement, he thought about it this way. So you have different species with different distributions across an environmental gradient, and that forms a community. So a community right here might have species 4, species 1, and species 2, but no species 3, whereas a community right here would have species 3, species 4, and species 2. And this is, again, something we see in nature quite a bit, where sort of the distributions of species can overlap quite a bit. and the resulting community is simply the sum of species distributions and their individual tolerances. So both of these concepts clearly have some merit, and we see them in nature. And some support for these come from more plant surveys. Guess what? Plants are sort of the main thing that we did community ecology with for quite a while. And these are the Great Smoky Mountains, where a guy called Robert Whitaker spent an awful lot of time putting up plots about the size of a classroom and counted vegetation and species in these plots. And so he literally went to different depth bands, hiking up the mountains, putting these plots down, and quantifying the species and their abundances. This is again what the table looks like. This one's actually turned around from what I would usually consider these tables to look like. So he has species on the as rows here, and then the different stations, as he called them, as columns, and then the abundances throughout. And he also visualized this in a way that looks very similar to what we saw by Gleason. So this is very clearly a community where we have differing species distributions across that elevation gradient, and they form these communities, right? So this would support Gleason's individualistic concept of ecological communities. Now the question is this a really good quantitative analysis of a community? Can we actually derive much from these curves? The answer is probably no. And luckily, in about the 1960s, we came up with this really good way of visualizing communities using something called ordinations. So this is a paper by Bray and Curtis, who were very active in this field. And something we can do are what's called a principal component analysis, or PCA, which you will learn to hate later on in this course. But essentially what it does, it takes a table like this and it turns it into something like this. So this is a multivariate ordination of these communities, where we have taken all the information, multiple sites, multiple species, so a highly dimensional data set, and we've compressed it into the 2 dimensions that tell us most about the differences in these communities, which in this case are called PC1 and PC2. So each of these little diamonds here is a different community, and the distance between them essentially reflects how similar they are in terms of their species composition. And lo and behold, what we can find here is that actually perhaps Clement wasn't all that wrong either, because we have these very distinct clusters of communities that perhaps, one may argue, might be more akin to these superorganisms that Clement was thinking about. So as a little bit of both, the PCA helps us see this. The other cool thing that we could do with the PCA then is we could use the position of each community on the PC axis and plot that, for example, against elevation. And we can see that very much they correspond to this elevation gradient that we've seen, that Whitaker covered in his surveys. So this is a really good way of describing patterns. So we can go out, we can survey something, we can use a multivariate technique to show, hey, these communities are different, right? And that would be an easy thing to do if we, for example, just want to say, what's the difference between these 2 communities, right? But it doesn't tell us much about the actual mechanisms that structure communities. And that's really where the money is, because describing something is fine, but understanding why it is a certain way is really much better. And that's what we're striving for as community ecologists. And so for most of the rest of this lecture, we're not actually going to deal with what I would consider communities. We're just going to deal with 2 different species. because at the end of the day, a lot of the mechanisms that drive community assembly is the interactions between species. And to keep it simple, we're only going to deal with two species. These guys right here are two gobies, two prawn gobies, they're called. The one on the left with the orange spots is Amblyeleotris guttata, or the spotted prawn goby, and the one on the right is Amblyeleotris wheeleri, or the gorgeous prawn goby. I think they're both gorgeous, but anyway. So gobies, just a quick foray here, are a family of very, very small fishes. They are distributed almost globally except for the poles. They come in all sorts of sizes, shapes, and colors. And there's about 2,400 species, I think, of gobies. They're my favorite fish family, and they're absolutely beautiful and adorable, and I love them dearly. Anyways, let's talk about these two gobies. So our assumption is that these two gobies, because they look very similar, they live in the same place, they need sort of the same resource, which is food and shelter. The shelter actually here is provided by these shrimps that dig little burrows, and the gobies will live in the burrow, and in exchange for getting housing, room and board, the goby will stand watch over the shrimp while the shrimp digs so it doesn't get eaten. It's a really cool symbiosis. But yeah, so these two species are in direct competition, and we can use models, mathematical models, to look at their population dynamics, and that can tell us something about how communities operate. There's going to be a little bit of math in the next few slides, but despair not, it should all be fairly digestible. Okay, so if we're modeling populations, we need a few different parameters in our model. So the first one is n, that is the population size. And then we're always doing this through time, right? Because this is dynamic, we want to look at processes rather than just patterns, so it is through time. So the subscript for our population size is at time t. So nt is the population at the time point t. So let's start with a single goby at n0, and that's 1. Okay, then at n1 we have n0 times 2, which is 2. At n2 we have n1 times 2, which is 4. You get the drill. We're going to 8, we're going to 16, and so on and so forth. Now that increase in population is our population growth rate, and that is denoted with the letter R. Our growth rate in this case, of course, is 100%, so we're doubling the population at every step, which means that our growth rate is 1, right? Percentage proportion, we're just going from 100% increase to 1. Okay, and if we do that, we get what's called the exponential equation. And let's just say we run that model for 50 years. The population of gobies will increase quite a bit. And as a matter of fact, in 50 years, we have about 900 trillion gobies, which is wonderful for everybody. Everybody loves gobies. But it's a problem for the gobies. Now, this may not be news to you, but populations can't grow infinitely, right? There's going to be something that stops them at some point. And that is what we generally call the carrying capacity. So the carrying capacity considers the competition for resources between individuals of the same species. So we're all competing for the same resource if we're in the same species. And K, the carrying capacity, denotes how many individuals can actually exist in a certain ecosystem of that species. So 1 minus nt divided by K tells us how far the population is away from meeting that carrying capacity. And that means the real growth rate of that population changes based on how far away that population is from the carrying capacity. And that gives us what's called our logistic equation, which is essentially just the population size at your certain time point plus the population size at the next time point times the growth rate, which is 1 minus the population size divided by k. And what that looks like is like this. So the population grows like crazy at first through the exponential growth curve. And then you can see it sort of slowing down a little bit until it actually meets its carrying capacity. And then it's stable right there. Okay, so far we've only dealt with one species. So I was lying earlier when I said we're going to look at two species. We actually only looked at one species. Let's bring the second species in, the gorgeous prawn goby. And we can do the same thing with that one. We're just going to set it at a slightly lower carrying capacity and a slightly lower growth rate. And what we get are two species that sort of, grow exponentially until they hit their carrying capacity, and that's it. What's wrong with that? Those two species are competing, and right now we're not letting them compete. Right now they just do their own thing. They're in their own tracks. They're not bothered by each other. But we can include something that's called a competition coefficient between those two species in our model. So that would then denote that there's some resource competition between those two species as well. So the idea here is that competition for resources is always most intense within a species. So an individual of the spotted prawn goby will 100% compete with an individual of the spotted prawn goby. The same is true for these gobies. They're 100% in competition within the species. And then among the species, competition is usually weakened because the niches might differ slightly. They might not fully eat the same thing, or they might prefer slightly different burrows. So competition is slightly relaxed, right? So we're essentially deriving here that the competition coefficient between those two species is 0.5. If we set this, don't be afraid of this equation down there. I'm not going to quiz you on it. But if we use that equation with that competition coefficient, we get something like this. So the first goby in green here shoots up, has a higher growth rate, higher carrying capacity. But then once the other goby gets off the ground in terms of population growth rates, this one actually dips back down below its usual carrying capacity because some of the resources are being used by this species right here, individuals of this species that have gone up and have grown in population size. I can also set an inverse competition coefficient. So now both of these species are having a negative effect on each other, and actually this species right here has might be a better competitor, so it actually has a stronger impact on this species. And you can see that it dips down and almost, results in the extinction of that goby in this system. But let's go back to even competition, where both of these species have the same effect. None of them is necessarily a better competitor than the other. And let's think about how this competition really works in the real world. So these two species compete for the same resources, and this sort of resource competition is usually thought of within the concept of ecological niches. So both of these species have niches that describe their use of different dietary items, their use of different habitats, space, time, you name it. One of them might be nocturnal, the other one is not, or something similar. And the important thing to remember here is that intra-specific competition, I mentioned this just now, so competition among individuals within the same species is always going to be higher than inter-specific competition, so competition between individuals of different species. Now, some of the most important figures in the concept of niches and their impact on competition are Robert MacArthur, who coined the limiting similarity theory, which means that species have to be sufficiently different in their niches to coexist stably. And also David Tillman, who coined the resource competition theory, which is essentially a way of hypothesizing that species species use different resources and have different limitations at different levels of different resources. Okay, so niches are all nice and good, and let's just envision these two species again. They have the same alpha, so they compete against each other at the same level. They have slightly different niches, so you should be good to go. But one of them just doesn't exist. It just basically conks out before anything ever happens. What else could be determining whether a species persists in this environment? The answer is it's the species fitness. And the fitness in this case is denoted as the growth rate. So if I set the growth rate of this species at 1.1 and the growth rate of this species at 0.1, sorry, you can see that, then I will find that this species dominates and basically monopolizes all the resources, and this species doesn't exist in the environment. And fitness differences can be derived from very different sort of ways in which a species can produce offspring. So it can be the growth rate, it can be the fecundity, it can be mortality or energy use. And most species have increased fitness when they are rare. Why is that? The answer is it is because there aren't as many individuals that use the same resources, right? So again, if intraspecific competition is always higher, then if you're rare in the environment, you should have less competition for those resources. This is what we call negative frequency dependence. Now, importantly, if two species are to coexist in the same place at the same time, then their niche differences have to outweigh the fitness differences. So if one species is only slightly different in terms of the resources they use from a different one, but has a massive fitness advantage in that they're just producing babies left, right, and center, and the other one isn't, then that species will likely outcompete the other one, and the other one will go extinct. And congratulations, you have just learned modern coexistence theory, at least in its basic sort of ideas. This theory was coined by Peter Chesson and then refined by Janneke Hillerys-Lambers, and it is sort of the underlying concept of how we conceptualize communities to exist and how we conceptualize species to coexist in time and space nowadays. Quantifying niches and fitness is a hell of a time. I've done a fair bit of it in my career. Niches, of course, you quantify through things like morphometrics. You can look at gut contents, resource use, and so on and so forth. Whereas fitness is often a little more difficult to quantify. You can look at population growth rates, of course. You can look at the growth rates of an individual. You can survey reproduction and other things. But yeah, there's some of the main things we do as community ecologists. And the conclusion here is that niches and fitness are the deterministic drivers of community assembly. So if our niches and fitness differences don't line up, then one species will outcompete the other one, and the other one will not be found in the community, right? But we can also observe cases where, for example, we have perfectly even competition, we have perfectly even fitness of two individuals, we have high carrying capacities, and generally these two species should be able to coexist just fine, but yet one of them doesn't exist. What happened? Well, the answer is that our local community is not isolated, right? They are always related to some of the processes we observe at regional and perhaps even global scales. And as a consequence, we often have things like emigration and immigration. So we might have individuals leave the local community, or we might have individuals coming into the local community. And for example, with our two gobies, we can envision a scenario where we have a reef that is downstream of a different reef. On that different reef, we only have, for some reason, the the spotted goby, and the spotted goby here produces lots and lots of offspring that gets washed to the other reef. So even though their growth rates and fitness are even, the immigration from a different reef actually results in the spotted goby outcompeting the gorgeous goby on that reef. And that can result in something called extinction. Sure, you've heard of it. Usually extinction is something we associate either with the dinosaurs or with some of the most emblematic sort of cases of large megafauna that have suffered from extinction due to human activity. But in reality, extinction is fairly common, at least at the local scale. So you get these situations where locally from a certain community, a certain species gets removed due to a variety of factors. And those factors can be fairly stochastic. So just think about, for example, this guy right here having a fondness for a certain type of goby and just collecting it all over a certain reef. So that's a fairly stochastic driver of local extinction for that goby. And those two processes, especially extinction and immigration, are the foundation of another theory of ecological communities that is quite prominent, that has quite a bit of relevance in this field. So essentially, this theory has these two processes, extinction and immigration, as its backbone. And it posits that these two processes alone will essentially govern how many species are present in a community, right? And the two most critical factors for those two processes are the area of a certain community that a certain community lives in and its connectivity. Why? Because if the area is very small, then the population sizes are likely to be very small. If population sizes are small, we are much more likely to see local extinction because it only takes the removal of a few individuals for the species to conk out. For connectivity, it's quite logical that that would influence the immigration part. If a system is really well connected, then it gets influx from a lot of different other places, and so immigration should be a lot higher. So then if we're looking at the interaction of these two factors, so we have large and small communities and we have well-connected and isolated communities, we can see that small and isolated communities have very low local species richness. We get, if they're well connected but small, we get higher species richness. And if they're really well connected and they're very large, we get the highest species richness. This is called the theory of island biogeography, and it was coined by Robert MacArthur and E.O. Wilson. And you might say, hey, wait a second, Robert MacArthur, wasn't that the guy who did the limiting similarity thing? Yes, he was. So beyond the niche-based work, he also was involved in this theory. And the theory of island biogeography has quite a bit of support. If you remember from earlier in this lecture, for example, estuarine fishes and coral reef fishes, some of the major drivers of species richness in these two communities were the estuarine area or the coral reef area and the connectivity. So very clearly there's something to be said for the validity of the theory of island biogeography. But if you've paid attention, then you might find that if it's really only about connectivity and area, then ecological differences don't really matter at all. So all the stuff we did about competition and niche differences and fitness differences doesn't seem to matter at all. And that's exactly what this guy said, Steve Hubble, who coined the neutral theory of biodiversity. And he essentially said that the only things that matter to determine the species richness of a community are speciation, extinction, and immigration. And he was quite successful actually in applying this theory to predict the species richness on coral reefs. And essentially what it does, it adds to this this theory right here, the theory of island biogeography, by adding speciation to the mix. So speciation in a system considers the sort of historical prevalence of diversification, of species speciating, and that will shift that equilibrium species richness that is determined by extinction and immigration along the x-axis to determine the actual species richness in the system. And as a consequence, essentially all that matters is the extinction, immigration, and what's called the regional pool of species. So this is what speciation in this case includes, right? And if that were true, then we should see a pattern where the local species richness is essentially completely determined by the regional species richness. So in other words, if we do this mathematically or in a graph, we should see that alpha diversity, which remember that's the diversity of a local community, on the y-axis is purely determined by gamma diversity. If that is the case, if that is true, then we should see a straight line here. However, if competition matters, if there's something about the species ecology, the competition, the niche differences, and the fitness differences, then we should see a divergence from this straight line. And this was what's called the regional biodiversity hypothesis, which was coined by Robert Rickliffs, which basically concludes our little history of different theories. This is not comprehensive by any stretch of imagination, but it's a good sort of vignette of what has happened in community ecology over the past 150 years. So we essentially went from just observing patterns to coining a whole lot of different theories, and they're mostly split into theories that basically say, well, this is all determined by the ecology of different species and their competition. So deterministic drivers such as niche differences and fitness differences, or the sort of camp that was mostly saying, well, this is mostly stochastic. It depends on the size of the community, the extinctions, and the immigration or the speciation patterns. And that's basically it for today. The homework for next class will be to read Lawton 1999, Are There General Laws in Ecology? And send in a 250-word statement as to whether or not you agree with the main message of the paper and why. The next class will be next week on Monday, February 3rd, and I look forward to seeing you all again then. Bye.